Hi, my name is Victor Dunagan. This summer, I had the opportunity of interning in the SenNet CUSP internship program at the Lee Lab at the University of Michigan. And the title of my project is Tissue Optimization for Spatial Transcriptomic Analysis with Seq-scope. So these are the topics that I'll be covering in my presentation today. I'll just be giving you a little bit of information on the background of this project, and then I'll move in explaining the experimental procedures and the results in future directions. So for a little bit of background on spatial transcriptomics is a molecular profiling method that is used for mapping gene expression in tissues. And initially, when this was first discovered, earlier methods were able to capture RNAs and see what genes were being expressed in the tissues, but they weren't able to do this as efficiently as they weren't able to sequence the entire genome within one experiment. So as methods began to advance, this is when we arrived at hybridization and sequencing and spatial barcoding. All of these methods use oligonucleotides planted upon a slide surface in order to capture mRNAs from tissues and then use this in order to obtain spatial information and genetic information. So now moving into Seq-scope. Seq-scope is the project which I worked on that uses spatial barcoding microarrays on repurposed Illumina flow cells in order to provide a more streamlined spatial transcriptomic process. And this is done by breaking it down into two main sequencing steps. And in the first sequencing step, the platform is sequenced and we're able to obtain the positional and the spatial coordinates of each of the oligonucleotides with their uniquely labeled barcodes, as you can see here. And then in the second sequencing step, once mRNAs have been captured from the tissues on the oligonucleotides, we're able to do a second sequence and obtain the genes that are being expressed in those locations. And the barcode in this instance is going to be referred to as the HDMI. This just means a high definition map coordinate identifier. And this is just a sequence found within the oligonucleotide in order to give identity to the spatial coordinates and the genes that are being expressed in different areas from the tissues. So I had two main research objectives in my internship this summer. I wanted to optimize the tissues in order to maximize the amount of mRNA that is being released and captured from the tissues in these experiments. And I also wanted to learn how to use the alignment operation in order to obtain the final product and the final image that was encoded with the genetic and spatial information. So now for a little bit on the optimization procedure. I started off by cryosectioning samples and then staining these samples in order to obtain an initial reference histological image. Once I was able to obtain these sections and do the staining, I started off by permeabilizing these tissues at different time points. And then after these tissues were permeabilized, the mRNAs were captured upon the oligonucleotides on the slide surface. And then these mRNAs were then used for reverse transcription in order to obtain a complementary DNA strand. And after we had the full product, which was the oligonucleotide with the captured mRNA strand and the complementary DNA strand, we enzymatically removed the tissues from the slide surfaces and then imaged the results. So this is the result of the tissue optimization experiment with mouse liver tissues. And so as you can see, we imaged them from, well, we permeabilized these tissues from time points from 0 minutes to 35 minutes. And the main thing that we were looking for here is we were looking for the time points that allowed for the peak fluorescence, as this was indicative of the mRNAs that were able to be captured from the tissues after permeabilization. And so we were able to determine that at the 15-minute time point, this is when we were able to obtain the most mRNA from the mouse liver. And for the mouse brain tissues, we were able to determine that at the 5-minute time point, this is where we were able to collect the peak amount of mRNA. And for the mouse colon, at the 30-minute time point, this is where we were able to determine the peak condition as well. Now that I was able to work through the wet lab procedure and obtain the mRNAs from the tissues, I now wanted to start by preparing the libraries for the second sequence so that we would be able to obtain the genetic information to go along with the positional information from this experiment. So as I said before, reverse transcription was done in order to leave us with the complementary DNA strand. And so then after we have that complementary DNA strand, we move into second strand synthesis and adapter PCR. And these two steps are done just in order to add on a unique molecular identifier to the strand, and then also add on unique primer pairs for sequencing in the end. And this yields us with the final product down here for the library structure for the second sequence. And as you can see, this sequence has the unique HDMI barcode that was printed from the original oligonucleotide so that the genetic information could be encoded along with the positional coordinates from the second sequencing reads. Now that the library products were obtained, we had to run a bioanalyzer trace in order to get an idea of what kind of library was prepared. And this is just so that we can send in this information with the samples as we get them sequenced. And so this was for the mouse brain samples. And for two samples here, we were able to achieve an average size of 782 base pairs and an average size of 749 base pairs. And this was the gel that was able to produce both of these traces here. And so now that I was completely done with moving through the wet lab procedure, I wanted to learn the alignment procedure to be able to arrive at the final image that contained the spatial information and the genetic information as well. So by using the original histological image and the spatial barcodes, we were able to construct a spatial barcode map using an algorithm that was made by one of the postdocs in the lab. And so first off, this algorithm starts by taking this information and segmenting the image into discrete squares, as you can see in this image, and it spatially builds barcode maps within each of these squares. And then after each of these segmented squares has been mapped, they're all paired together within a single image to produce a spatially barcoded image. And then we move on with a SMATCH method, and the SMATCH method is an algorithm that goes back and it checks the first sequencing products to be able to make sure that the HDMIs or the barcodes can be found among the barcodes within the spatial barcode image. And then this is printed and makes the SMATCH image. Then the SMATCH image, along with the histological image, goes through two more steps in order to arrive at the final aligned image. And so we basically align the HDMI reads to the reference genome, and then we're able to produce a spatial gene expression matrix as well that reads and has the information for the HDMI, the positional coordinates, and then the gene that's also been sequenced. So as far as future directions for this work, the main goal of the NIH SenNet consortium is to map senescent cells throughout the human lifespan in order to understand physiological health. Cellular senescence is the phenomenon in which cells lose their replicative potential as time goes on. And this is due to the fact that as DNA continues to replicate, the telomeres at the end of the DNA continue to shorten over time, as you can see in this figure over here. And this leaves the DNA vulnerable to damage, and this prompts the DNA damage response. The DNA damage response then leads to cellular senescence, where cells stop dividing in order to stop the propagation of damaged DNA. And this leads to a variety of negative effects, such as arthritis, cancer progression, cardiovascular disease, and inflammation, just to name a few. So by perfecting this process with Seq-scope, other labs in the consortium will be able to use this application in order to further study and characterize senescent cells and understand how they can be applied to different pathologies. And in closing, I'd like to give a special thanks to my PI, Dr. Lee, my mentor, Dr. Kim, members of the Lee Lab, the NIH SenNet CUSP internship program, and the Michigan SURF program. All of their support and mentorship allowed me to obtain a deeper understanding of the project and learn new lab skills as well. Thank you.